In this video, I'm going to be discussing the Krebs cycle, which is the third part of aerobic respiration, and it's the fourth video of the respiration stuff that I've done. So I've done an introduction, glycolysis, the link reaction, and the Krebs cycle. So if anything that I say is a precursor is a bit confusing, go back and watch one of those videos. I moved the Krebs cycle over here because it's a big cycle, and I want to make sure that I can get it all in because there's quite a lot that comes into a lot of products coming in and out. But basically, the Krebs cycle produces loads of coenzymes and a little bit of ATP. That ATP is going to go on to be energy, and the coenzymes, again, like all the other stages, are going to send reduced NAD and a few others to oxidative phosphorylation, which is the last stage of respiration. All this takes place using a series of oxidation and reduction reactions within the mitochondria matrix. So again, this stuff is in the introduction to respiration. It's quite a big topic made up of lots of little bits. So it's a good idea to keep referring back to that video if you get lost. So in the last stage, the link reaction, we produced acetyl coenzyme A. Like I say, if you haven't watched this video, or the previous video, go back and watch it because it's going to help you a lot. So, coenzyme A, and that had 2 carbons. So we end up with 2 carbons. What's going to happen, the Krebs cycle is a cycle, so you're going to see something like, if you've done the photosynthesis and the Calvin cycle, you're going to see a product that's just appeared out of nowhere, but at the end it's all going to make sense. So this acetyl coenzyme A joins with a 4-carbon compound called oxaloacetate. Now, some specifications write it like this, and some just say a 4-carbon compound. So I'll just write 4-carbon. The AQA syllabus uses this name. This is already in the cycle, and you'll see how it's produced in a minute. But basically, the acetyl coenzyme A joins with that, and that produces a 6-carbon molecule. A 6-carbon molecule called citrate. And this, you would say, is step 1. It's the formation of this citrate, citric acid. The coenzyme A, as I said in the last video, its job is basically to carry this over. So once it's done its job, the coenzyme A leaves and goes back to the link reaction. So we're just left with this, and they combine to make this citrate. And that's step 1 of the Krebs cycle. Step 2 is the formation of a 5-carbon compound. And strangely, no specification within the UK gives it a name. So I'm not going to tell you the name because there's no need to know. So a 5-carbon compound, and hopefully you already notice, it's a wonky line, you already notice that it's a 5-carbon compound, but we started with a 6-carbon compound. So that must mean we're going to lose a CO2 through decarboxylation. That's a bit of a tongue twister. So we'll say we've lost 1 carbon there. So now we end up with our 5-carbon. And now we get our first time we see in this stage oxidation of this product. And when it's oxidation, something's going to get lost from this product. Our NAD is going to come in, and when this product loses something, this gains, and when you get a gain, it's called reduction. So we end up with reduced NAD, and this leaves and goes somewhere else, which I'll explain in a minute. So we had step 1, formation of this, and we got step 2, the formation of the 5-carbon compound. I'll actually write that 5-carbon compound. Now, people get this cycle confused within photosynthesis, because here, what you would see in photosynthesis is a product coming out. The Krebs cycle, remember, in the first slide I said, makes loads of this, an ATP. So you don't get a carbon-based product at the end. What happens now is this 5-carbon compound gets regenerated back into this, and the cycle's going to start. And along this long line is the little coenzyme products that we make. So the first thing that happens is that you can see is that we're going to lose another CO2. Told you it was a squeeze. We lose another CO2, which means we've got another carbon gone. Means we've only got 4 carbons to play with, and that's how this ends up here. Now, remember, you can call reduced NAD NADH. It's the same thing. I'm going to try and squeeze reduced NAD on here. So what we're going to get now is NAD is going to come in. So it's a series of oxidations. And you get your reduced NAD. Reduced NAD leaves. Told you it was a squeeze, sorry. So we get one product of reduced NAD leaving. We also get some ADP plus this inorganic phosphate coming in and transforming it, and this gets joined together, which makes the product of ATP. You also get another one of these reactions after that. So your NAD comes in, and you're going to get reduced NAD out. And then lastly, you get another thing, another coenzyme called FAD, which for the sakes of this video is like NAD. And again, that becomes reduced FAD. And what I'm saying when it becomes reduced is that it's taking hydrogen from here. So when you're getting reduced, it's carrying a bit of hydrogen around. And all that's going to make sense in the next video. So I told you there was a lot squeezed on. In some pictures, you will see all this the same here. But rather than reduced NAD being written twice separately, sometimes you'll see like 1 CO2, 2 reduced NAD, 1 ATP, and 1 FAD. And if you see that, that's basically what it means. But with the majority of the specifications within UK, it talks about the reduced NAD appearing twice. So that's why I've written it in that way. Remember that we had 2 of these products. So this whole cycle goes around twice per 1 glucose molecule. So all this is going to happen. There's going to be another one happening with the other lot of acetyl coenzyme A. So let's just talk about the products again. We've got our coenzyme A. Is important to remember that leaves the cycle, so it's a product. And that leaves the cycle to go to the link reaction. Another product you get is oxaloacetate at the end, but that stays in the Krebs cycle. But it's still a product. You also get 1 CO2 lost here, and you get another one here, so that's 2 CO2s lost. You gain 1 ATP, and you gain 3, 1, 2, 3 reduced NAD, and you gain 1 reduced FAD. That's where they all come. So you've got, but it happens twice, so I'll write it all per glucose. So you're going to end up with your 2 coenzymes A, which are going to go to the link reaction. You produce, we'll go in order, you produce 2 lots of ATP, which will be used as energy. Remember, I've only said one here, but it happens twice. So 2 ATP, you're going to produce 4 CO2 as waste. You're going to produce 6 reduced NAD, and that's going to go to oxidative phosphorylation, and you're going to produce 2 reduced FAD, and that's also going to go to oxidative phosphorylation. Okay, I hope all that makes sense. It's quite confusing, I think, with all the little chains, and in some books they have slightly different pictures and so on, some videos. But these are your products, and it's important to remember now for when you start adding up how much energy is made from one glucose molecule overall. And we need to have a good grasp of what these are and where they're going to go in the next video. Okay, thank you.